have this, we do this AGN thing.
Mhm.
You know, let me so it's not the fungal, it's the inflammatory autoimmune kidney disease, just to give you an overview of things, and you can read our papers. So I thought we'll present this in the, but not enough time. So this is a called antibody mediated glomerulonephritis, AGN. Okay, so what happens is, this happens in many autoimmune diseases. So autoimmune disease is that when your immune system damage your own body. Arthritis is one of them, psoriasis is one of them, lupus is one of them, in which in all these cases you have antibodies generated in your body that recognize your own body cells rather than, okay, and in all these patients you do see that there is kidney pathology, whether it's ANCA-associated glomerulonephritis, each anti-neutrophilic cytoplasmic associated antibody associated. So here the antibody is generated against proteins expressed in neutrophils in your body. And these antibodies, this is the glomerular slide, this is where your blood gets filtered, and this antibody gets deposited here, that initiates an inflammatory reaction, and that damages the kidney. This is a disease known as Goodpasture disease, where you have antibody that recognize the glomerular basement membrane, and it gets bind here, deposited here, that initiates inflammatory reaction, damages the kidney. This is known as lupus patients, lupus nephritis, where you have antibody against your own nucleic acids like DNA, RNA, that gets deposited in the glomerulus, initiates the process and inflammation that damages. So you can see there is common thing, right? You need antibody to process, but the antibody is not against bacteria or fungi or pathogens, it's against your own body cells. The thing that differs is the antigen. Here it's against neutrophil proteins, here it is antibodies against glomerular basement membrane protein, and here it is against DNA and RNA.
Different type of.
So the procedure is the same, the pathogenesis is the same, but the antigen against which the antibody is generated is different. So we try to study this in our model in a mice, and we have a not similar thing but close to that, this in mouse. So what we do, we take black 6 mice and immunize these mice with rabbit IgG, immunoglobulin G, in adjuvant, complete fluid adjuvant. Idea is, as rabbit IgG is a protein that the mouse never saw, if you immunize with rabbit IgG, the mouse will develop antibody against rabbit IgG. So it will have mouse antibody that will recognize rabbit IgG. Got the point?
Yeah.
And then 3 days later, we inject mice with anti-GBM, glomerular basement membrane antibody. That is raised in rabbit. Means we isolated glomerular basement membrane from mouse and immunize rabbit with this glomerular basement membrane. As glomerular basement membrane is a mouse antigen, in rabbit it will generate rabbit anti-glomerular basement membrane antibody. Now this is a rabbit antibody, and now you have mouse antibodies that is recognized as any rabbit antibody. So this antibody will go and bind, as it's against glomerular basement membrane, it will go and bind here, and the mouse antibody that recognizes the rabbit, it will form immune complexes, means the antibody will keep on binding, and this will initiate an inflammatory reaction, and our lab showed the importance of interleukin 17 in this process to cause kidney damage.
Okay.
So this is the way we model this thing. Do you understand?
Yeah.
So you have to understand this. So you immunize mice with rabbit IgG so that mice can develop antibody against the rabbit IgG. Then you come back with intravenous injection of rabbit anti-GBM serum, that is antibody against mouse anti-GBM. As because this is a rabbit antibody, now this antibody generated against rabbit IgG will bind here, and this will form an immune complex that leads to inflammation of the glomerulus.
Yes.
And the way we measure kidney dysfunction is through serum blood urea nitrogen and inflammatory influx of cells in the kidney.
Okay.
Is it clear?
Yeah.
But if you read, so this is the first paper where we did, as I said, we measured kidney dysfunction. Wild type mice, IL-17 receptor knockout mice. So you can see the blood urea nitrogen is much lower in comparison to the wild type mice. Means they are protected. If you don't have IL-17 receptor, they are protected. And you can see less inflammation in the glomerulus, and the glomerulus is completely damaged. If you treat mice with anti-IL-17 monoclonal antibody, as IL-17 is bad, if you neutralize IL-17, you see the mice is protected. There it is. Can you see? So IL-17 is important in the pathogenesis of antibody-mediated glomerulonephritis. Okay? And in this paper, if you read this Kritikas paper, how IL-17 works, because it produces cytokines and chemokines that bring neutrophils in the kidney.
Yeah, IL-6, IL-1.
And this damage is basically not caused by IL-17, but it's the neutrophils that come there and cause the damage. So in this paper, what we did is, we wanted to see how neutrophils get energy to cause the damage. So if you can block that, probably it's good to kind of prevent glomerulonephritis. We know neutrophils require glucose, right? So we induced AGN in mice, and then we get on neutrophils, CD11B by, Ly6G, and this is a 2-NBDG. 2-NBDG is nothing but a glucose analog, fluorescent glucose analog. So it will be green. So we inject it in mice 30 minutes before you sacrifice the mice. And then you sacrifice the mice, look at the neutrophils that are in the kidney at day 7, this is the early time point, and day 14, it's the late time point. And you can see in comparison to no AGN, where there is very little neutrophil which don't take up much glucose, now you see neutrophils have taken up 2-NBDG, which is a glucose-like molecule. This will suggest that in the nephritic kidney, in the AGN kidney, neutrophils infiltrate and they take up glucose. And this is where you see in non-AGN kidney there is very little glucose uptake, 2-3%. Whereas in kidney AGN there is 17-15% of cells take up glucose. Now we know neutrophils has two. Now we know neutrophils, you need glucose transporters to take up glucose. And neutrophils have two glucose transporters from De Dong's paper, GLUT1 and GLUT3. So we wanted to see whether the neutrophils that come in the kidney, in AGN kidney, express GLUT1 or not. So what we did, we stained for GLUT1, and you can see in comparison to the no AGN mice, there is increased expression of GLUT1. Means neutrophils upregulate GLUT1 and they take up.
Okay, just for GLUT1.
Yeah.
Okay.
And we also measured, I don't have the figure here, but we can go to the figure directly. This is the figure 1. So this is what I told you. And then we sort neutrophils, flow sort neutrophils from the kidney and measured the GLUT1 and GLUT3 expression, mRNA expression. And you can see GLUT1, the white column. Now, the GLUT1 is upregulated after AGN in comparison to no AGN control, which is white column. But GLUT3 is not. GLUT3 is kind of remains the same. Although it is higher expressed, constitutively higher expressed, but it's don't change. And when you look at the protein expression, you do see that there is higher protein expression in comparison to the no AGN control kidney. Now, protein expression is different, but GLUT need to go on the surface of the cell to take up glucose. So that's what we did, whether after GLUT1 express, this is an image stream analysis where you stain just the way you do flow cytometry. You stain neutrophils, you stain for GLUT1, okay? And then what you do is, instead of running it in flow cytometer, you run it in Amnis image stream analysis. So it's a combination of microscope and flow cytometry. Here you can see where the protein, it's not only how many cells express GLUT1, but it also gives you where is the protein expressed. Mostly GLUT is in the cytoplasm, as you can see in the middle. Now when you get activated, it is now transported on the cell membrane. So it's not that GLUT is upregulated, but it also translocation of GLUT1 occurs on the cell membrane of neutrophils, and this is shown here. This is the percentage GLUT1, Ly6G is the red Ly6G that you use. It actually makes the boundary of the neutrophils so that you know exactly that the GLUT1 actually moved to the cell membrane of the neutrophils. Okay?
Okay.
I'm just explaining you so that when you will read the results, you will understand. Now this is the actual experiment we'll start. Now we'll determine whether GLUT1 is important or not. Now the problem of working with, there is no global knockout mice of GLUT1, because GLUT1, if you do a global knockout mice, homozygous knockout mice, they die. But what we did is, we crossed it to ERT2-CRE. So this is a GLUT1 flux mice, SLC2A1 mice, which we have in our facility, right?
Yes.
We crossed it to ERT2-CRE mice. So ERT2-CRE mice is the mice that responds to tamoxifen. What happens is, all cells express the receptor ERT2 that responds to tamoxifen. So if you give tamoxifen in the ERT2-CRE positive SLC2A1 flux mice, what happens is, because of the tamoxifen binding to the ERT2-CRE, it deletes the GLUT1 gene. This is called conditional knockout mice. Okay. In this way, you can make a conditional global knockout mice, right? Basically, yes. Because ERT2 is expressed all in your cells. In this way, you can avoid the problem of homozygous GLUT1 knockout mice to die at a very early age. So ERT2-CRE positive SLC2A1 mice will survive nicely. Now when you have to do the experiment, you want to delete GLUT1 in the mice, you inject tamoxifen. And it's because mice is adult, now deletion of GLUT1 does not affect.
Okay.
And the control is ERT2-CRE negative SLC2A1, means there is no CRE. So this will not delete. So mice with deletion of GLUT1, mice without deletion. And what you do is basically you inject tamoxifen for 4 days, from day -6, -5, -4, -3. Then you give anti-rabbit IgG plus CFA. Then you come back with anti-GBM serum. And then you assess how these mice are doing in terms of AGN. So this actually shows in the kidney when you inject tamoxifen, ERT2 positive flox mice, it's deleted. SLC2A1, GLUT1. Okay? Now, at day 14, you assess how do these mice do. In comparison to the control mice, that where the GLUT1 is not deleted, you can see there is a significant reduction in the blood urea nitrogen level. Means when there is no GLUT1, you have less kidney disease, less kidney dysfunction. Similarly, serum creatinine, in comparison to the wild type mice, when you don't have GLUT1, these mice are protected. Means GLUT1 is important for the pathogenesis of AGN. Any questions?
So it means if we delete the GLUT1, the mice is not more susceptible.
Yes,
Okay.
mice is protected. So you need GLUT1 expression for the pathogenesis, for the inflammation. So this is just to support immunopathological. This is a PAS staining. This is no AGN control. This is after you do AGN. You can see that glomerulus is swollen, big in comparison to the less severe. And this is the measurement of the crescent formation. These are the kidney pathological features. You have crescent formation, abnormal glomeruli, and tubular inflammation score. That you know that in Candida you don't see these things because Candida don't go in the glomeruli. It's in that, and you have tubular inflammation, right? You see neutrophil coming in. It's the same thing. Now, as because we have these mice, so here we deleted at the beginning of the experiment. So basically from day -3, it's a global knockout mice, right? It didn't have GLUT1 from day -3. But you can always ask at what time of the pathogenesis is this GLUT1 important? Basically, as because like during early time point or during the late time point. You understand?
Yeah.
Because you can delete the gene by 4 injections of tamoxifen at different time post AGN. Like at day 0 when you are giving anti-GBM serum, then you inject this. Basically you are deleting at an early time point and see how the mice is doing in comparison to where you delete at late time point. This will tell you at what stage of the disease this GLUT1 expression is important for kidney pathology.
Okay, so which one is, we can say which one is more exact time for using GLUT1?
Yeah, which at what, not exact time, means at what stage GLUT1 is important.
Okay.
Is it like you have a disease, right? You have early time point, like when you give Candida, 24 hour is an early time point,
Okay.
96 hour is a late time point, like as you progress. So similarly during the path, it's a 14-day period, and you actually give early time point. If you block the thing in the early time point, if you delete the gene in the early, what happens? Or if you delete at the later time point, what happens? What we found is that if you delete the gene at the early time point, this is your control mice, the mice are protected in terms of BUN. But if you delete the gene at later time point, like day 9 to day 12, they show comparable expression, indicating that GLUT1 expression during the early few days after anti-GBM serum injection is important for the pathogenesis.
Yeah.
Did you understand the experiment? This tell you actually the time where GLUT1 is important. Now we wanted to know, we answered time. Now we wanted to know source. Because GLUT1 is expressed in your immune cells, it is also expressed in your non-immune cells. And kidney is made up of both neutrophils, monocytes, macrophages. These are all immune cells that infiltrate. But kidney has also epithelial cells, fibroblast cells, mesangial cells, which also express GLUT1. In GLUT1 global knockout mice, you cannot distinguish, right?
Yes.
Because it is deleted everywhere. Now, to understand the contribution of hematopoietic cells, which are immune cells, versus non-hematopoietic cells, which is the non-immune cells like epithelial cells, we did a bone marrow chimera experiment. Basically, what you do in a bone marrow chimera, you irradiate them. You get rid of, when you irradiate them, what happens is all your immune cells die.
Yeah.
And then you reconstitute those mice with either wild type CD41 control cells or PMN delta GLUT1 cells.
So this one is one sub project. This one is
No,
project.
this is done before once.
Okay.
This has been lying, we didn't publish. PMN delta GLUT1 mice. Wansok never worked with GLUT1.
Oh, yes. Yeah.
CD45.1 and 45.2 are known as congenic markers by which you can differentiate between your control bone marrow cells and CD45.2 for PMN delta GLUT1 cells.
Yeah.
Okay? And then what you do is following. We irradiate, then you have to wait for 8 weeks for the mouse to fill up with these cells.
8 weeks after irradiation?
Yeah. After irradiation, you have to transfer these cells in these mice. And then allow for 8 weeks. Like if you transfer control CD45.1 cells, this mice will be full of CD45.1 cells. If you transfer CD45.2 cells, this will be full of CD45.2 cells. So basically now, the idea is when you have a donor mice, means recipient mice, which is wild type, and you have transferred donor bone marrow cells from this mice, this will behave like a wild type because both hematopoietic and non-hematopoietic cells have GLUT1 expression. In contrast, ERT2 Cre flocks, SLC2A1 flocks, this mice will serve as a negative, positive control, where all the hematopoietic and non-hematopoietic cells express GLUT1.
Okay.
Right? Now, when the recipient mice has lack GLUT1, means epithelial cells, non-hematopoietics, and you transfer them with immune cells that has GLUT1. You understand this group?
Okay.
And conversely, wild type mice, if it's recipient, that means your non-immune cells like epithelial cells and other cells will have GLUT1, but you transferred ERT2 Cre SLC2A1 flocks mice, means your immune cells do not have GLUT1.
Okay.
This is how bone marrow chimera is made. So basically you are getting one compartment has GLUT1, other don't have. And then you see whether if GLUT1 is important for disease pathogenesis in non-hematopoietic cells or hematopoietic cells. And what we found is that when you measured blood urea nitrogen, when GLUT1 is not expressed in the immune cells, they have less disease.
Okay.
In comparison to here, it's almost equivalent because GLUT1, when GLUT1 is absent in non-hematopoietic cells like epithelial cells, there is no change in the disease. It's not important. But where it is important? In the immune cells. So these are the parameters that we measured. So finally we come, now we say that GLUT1 is important in the immune cells. So which immune cells? Neutrophils is very important for the pathogenesis of AGN, right? And neutrophils also express GLUT1 in the figure 1, and they actually take glucose using the GLUT1. So we generated MRP8 Cre mice to GLUT1 mice to generate PMN delta GLUT1. So where GLUT1 is now specifically deleted in neutrophils, not everywhere. And when we tested these mice for kidney dysfunction after AGN, you can see these mice have less BUN in comparison to control mice, indicating that GLUT1 expression is neutrophil is required for kidney pathology and inflammation in AGN.
Okay, interesting.
So we also measured kidney injury marker, this kidney injury marker, and there is less expression of kidney injury marker because there is less disease, and these are the parameters. So next we try to assess why is this mice do not develop disease. One thing that you should realize that when you immunize with there are two things happening here. There is a systemic immune response in terms of B cell response, antibody response is required, and there is also kidney specific inflammatory response. Both these things are important for the disease to happen, right?
Yes.
So you don't know when you delete GLUT1 in neutrophils whether it is affecting this systemic immune response or is it affecting the local inflammatory response. To measure that, because GLUT1 deletion in neutrophil minimize disease pathogenesis of AGN, right? Mm-hmm. So first we assess the systemic immune response, and the way we can do is measure the B cell response. B germinal center formation after immunization in PMN delta GLUT1 mice. So basically we immunized control mice and PMN delta GLUT1 mice, means neutrophil specific GLUT1 knockout mice, and measured germinal center B cells, and we see that there is similar germinal center formation. We measured plasma cells with the antibody forming cells, we see similar plasma cell number. We also measure the total antibodies like total IgG, IgG1, IgG2A, which are generated after immunization. There was no difference. We measured T cell response, which is important, like activated CD4 T cells, interferon gamma positive CD4 T cells, IL-17 positive cells in the spleen after immunization, and everything is normal. Telling this data tells us that neutrophil specific GLUT1 expression is not required for systemic immune response generation. Then how is it still affecting the disease? It is because it is affecting the local inflammatory reaction in the kidney. To test that, we made a mixed bone marrow chimera mice. One of the problem of GLUT1 study is that if you have less inflammation or more inflammation, the amount of glucose will be different.
Okay.
So to kind of do a head-to-head comparison in the same mice, so that the cells will see equal amount of glucose, we did a mixed bone marrow where we irradiated mice, made it an empty mice, and then instead of separately transferring cell, we transferred control or PMN delta GLUT1 cells together in 1:1 ratio, allow it to reconstitute. Now these mice will have half of the cells control cells and half of the cell will have PMN delta GLUT1 cells. Then we give AGN and measured neutrophil infiltration in the kidney, apoptosis, cytokine, chemokine production, ROS production, all of which are important for the kidney damage. We see that not in the early time point like day 7, but in late time point there is a difference in the neutrophil infiltration. So mice which don't have GLUT1, they don't really, is less in AGN mice. Similarly, monocytes and macrophages are also less, but not in the early time point. When we measure cytokine and chemokine expression, we do see again, at late time point you see that there is a diminished cytokine and chemokine gene expression like CXCL1, CXCL2 and CXCL8. In terms of inflammation, it affects the late time point, not the early time point. Then we also measured tissue damaging molecules, right? Neutrophils can damage tissue through ROS production, through MPO production, through metalloproteinases that degrades the extracellular matrix proteins of kidney. And we do see that although there is no difference in the cytokine, chemokine genes and early time point, here you already see that there is a difference. They produce less ROS, although not MPO, and they also produce less MMP9, which is a matrix metalloproteinase 9, matrix metalloproteinase 2, and LCN2, lipocalin 2. All these are tissue damaging molecules. So the absence of GLUT1 in neutrophils does not affect the inflammation at the early time point, but they affect their tissue damaging function by production of ROS, MMPs, and lipocalin 2.
Okay.
Finally, the last figure is we have inhibitor that can inhibit GLUT1 expression, WZB117. Can we actually treat mice with WZB117 and reduce tissue pathology in AGN? And that's what we did. We treated mice either with PBS or WZB117 and see how do they do in terms of kidney pathology. And you can see when AGN mice are treated with WZB1, they have less kidney pathology, less kidney damage as they express less KIM1, and also kidney inflammatory parameters are also compromised, indicating that blocking GLUT1 expression, although it blocking everywhere, you see that GLUT1 expression in kidney or GLUT1 blocking have beneficial effect in terms of kidney diseases. And these are the neutrophil infiltration in treated versus all of them got reduced. Similarly, we actually kind of try to time out based on our data of, remember the data where we figured out which time point is important, early stage or late stage. So we did the same experiment, but instead of deleting them with tamoxifen, now we used WZB117 blocking during the early time point and during the late time point. And again, we saw that during the early time point, it is important. If you block GLUT1, during the initial few days, you can block the disease, but once it is kind of past the early stage, the neutrophils are probably, you have already infiltration of cells that is not responding to the treatment.
Okay.
So this is where the paper will be, and I have most of the data worked out. A few things you can see I have to put the figures. I've also wrote the results section of the manuscript based on these figures. And the method section and part of the introduction section. I will give this to you for you to read, and also read all our papers, like Kritika's paper, Didong's paper, who worked on AGN system, so that you understand the.
Yeah.
I've already done most of the things, but I couldn't finish it because there's too much work going on. I hope to come back and finish and submit.
Okay.
So do you want me to give a printout, or
Yeah, give me a print.
and with the
Well,
figures?
Yeah. It's easier to figure out the story.
Yeah, okay.
Or
But
send me an email.
I cannot email you the PowerPoint because that will
Yeah.
be too big.
Yes, yes. Okay. Just, is it, if it's possible, just give me a basic figure to understand the.
Yeah, this is already the figures, right?
Okay.
These are the final figures that will go in the
Okay.
paper. I will give you this, but I cannot send you by email.
Mhm.
That's what I'm trying to say, is that these are very big files.
Yeah.
But it
No,
will
I
not.
see, yeah.
I can print out everything and you can read. Next week will be to read everything about AGN and also the paper, so that you understand.
Okay. So just give me the text.
Yeah, I'll print out and give it to you.